Good morning, everyone, and welcome to today's MPG Primer. We're very excited to have everyone here today for our very special speaker, Dr. Heidi Rehm. She's an investigator in the Center for Genomic Medicine at MGH, co-director of the program in Medical and Population Genetics at the Broad Institute, as well as medical and clinical lab director at Broad Clinical Laboratories. She's also the chief genomics officer at MGH and professor of pathology at Harvard Medical School. Her wide-ranging work is focused on discovering causes of rare disease and building resources to support the integration of genomics into medical practice. We are very lucky to have her here today to speak about the clinical interpretation of genes and variants for disease causality, which is such an essential part of translating all of our genomic discoveries to patients. So thank you so much. Dr. Rehm is very happy to take your questions during the talk, so please either type in the Q&A and we'll monitor for that, or come up to the mics and voice your question. Thanks.
Thank you. Great to be here. And I actually remember to load up my slides to a Google link, and it whoa, it's got a lock on the top. I guess I'll just go put this over here. There's no flat surface up there. Okay. So I am happy to send my slides out afterwards if anybody wants a copy of them. And as was said, I'm happy to take questions along the way. I can move this a little closer. Okay. All right. So some of the context of this topic that I'm going to talk about today around interpretation or classification of genes and variants is really housed in the context of a large NIH-funded program that I've been part of now for 10 years called the Clinical Genome Resource. And it's really a whole infrastructure that we'll talk about to really bring together evidence, to use it to curate both gene disease validity, is the gene associated with a particular disease, to curate variant pathogenicity, to curate dosage sensitivity, is loss of a copy of a gene or gain of a copy of a gene pathogenic for a disease, or clinical actionability. If I find a pathogenic variant in this variant and give it back to a patient, is there some action that could be taken to improve health? So those are the major domains that ClinGen focuses on to curate information, ultimately to disseminate it and improve clinical care, as well as research. There's over 2,000 people involved in all of this work around the world, and it's largely funded through 3 core grants, as well as now over 20 different disease-focused grants from different NIH institutes. Okay, so and some of the resources and the way we distribute the curated knowledge. So we're doing a lot of the work over in ClinGen, and that work is on our website, clinicalgenome.org. And we do expert curation using a variety of interfaces: variant curation interface, gene curation interface, dosage curation interface, actionability curation. You see a theme here. And then the somatic work is done in a separate database called Civic through a collaboration with the somatic cancer community. And that's where all the expert curation happens. But there's 2 other databases that we work closely with. One is ClinVar, that's supported by NCBI. And that is a place that ClinGen puts all its expert curated classifications into. But so can anyone else in the world share their own classification variant. Anyone can submit to ClinVar. In addition, we've recently launched a database called GenCC, which I'll talk about briefly. And that is a place that anyone can share their own classifications of gene disease relationships. So I now call this the ClinVar for genes sort of thing. And we support that project as well. So I'm going to talk about a little bit about the distinction between classifying genes versus variants. And actually this original figure I stole from Daniel MacArthur, because I thought it was a great illustration of the concept of having gene level evidence on this axis and variant level evidence on this axis. So there's lots of variants, like the common Delta 508 variant in the cystic fibrosis gene, CFTR. So that is both pathogenic, well-established, and the gene is well-established as associated with cystic fibrosis. So lots of evidence on both dimensions. Of course, in every gene, whether it's well-established for a disease, we constantly find variants of uncertain significance. So that is common, very low variant level evidence, despite the fact that the gene is clearly associated with disease. Of course, there's lots of variants throughout our genome in genes we don't understand. So the goose and the goose, as some people call it. However, this category can't exist. You cannot have a variant that you label as pathogenic if the gene has never been associated with disease, because the term pathogenic is causal of disease, pathology. And so if the gene hasn't been implicated in disease, you can't call the variant pathogenic. You could say, it looks damaged, it looks like it doesn't work, it's deleted, you can just say that's probably not functional, but you can't call it pathogenic, which implies an association with disease. So it's important to do the gene classification first before you then start classifying variants. So I'm going to talk about this sort of gene disease validity element. In terms of the whole catalog of all the genes that have been reported to have a role in disease, many of us use the OMIM database, and you can go to the statistics page and get the stats on at any point in time and how many genes. And I just summarize them there. 24% of the estimated number of genes in the genome have been reported to underlie a Mendelian phenotype. And mostly today, we're talking about Mendelian causes of disease. But there is evidence for over 10,000 more genes being involved in disease. And that comes from a nice paper that both combined human data from the NOMAD database, looking at gene constraint, so genes that seem intolerant to loss of function variation, as well as mouse models that show a phenotype, and lots of human phenotypes recapitulate, or mouse phenotypes recapitulate human phenotypes. So combining that data suggests there's anywhere between 5,000 to 10,000 more genes to be implicated in Mendelian disease. So we still have a lot of work to do. So, however, all of these genes that get published as disease genes, they don't always have sufficient evidence for that claim. And so one of the projects that ClinGen has been involved in is really systematically defining the evidence for those genes and their role in disease. And we do that through looking at both genetic evidence, so case observations with phenotype, segregation in large pedigrees or small pedigrees, case control studies. We also look at experimental evidence, expression of the gene in specific tissues, model organisms, rescue studies of the function. And by aggregating that evidence together in a systematic sort of semi-quantitative approach that's been published in this paper, we then classify each gene with respect to a given disease according to these strength levels. That work then can be used in clinical care, because this paper that was written by a working group of the American College of Medical Genetics, Genomics, defined what level of evidence is necessary to put a gene in a disease-focused multi-gene panel. So, and that is defined as moderate and above can go in a gene panel, versus all these limit and below should stay out of the disease-focused panels, but obviously they may be looked at when you're doing exome and genome sequencing, where you're looking at everything. So that's sort of how this framework then ties into how we use it in clinical care. And I mentioned this framework is semi-quantitative, so these different evidence categories can garner up to 12 points for genetic evidence and up to 6 for functional data. And that can get you to strong. And then in order to move from strong to definitive, you need to see replication of the finding and standing the test of time, which means at least 3 years out there, time for things to be objected to. And so this is how this, sort of scoring and classification work happens. And in our expert panels, we'll have biocurators curate all the evidence, present it to the experts, and how they scored each of the points. The experts will then weigh in and say, gee, I wouldn't give that mouse model the full 2 points because it doesn't really recapitulate the phenotype, because I'm an expert in this phenotype. So sometimes our biocurators might not quite know how to evaluate a particular animal model, whereas the experts who really understand the phenotypes can weigh in. Or you scored, points for those patients, but I don't think they have the true phenotype. I wouldn't score those, so that's kind of how our experts weigh in on this stuff. And then ultimately at the end, a final classification is approved by the expert panel. So that work has been done across 2,350 gene disease relationships. And if you go to our website, this is just screenshots from the clinicalgenome.org website. You can see the distribution for any disease area that's been focused on. But the summary of where we are with the 2,350 gene disease curations that have been done to date can be seen at the top in terms of how many are in each category. And the take-home message is that about 23% of claims in the literature for gene disease relationships are insufficient to support that claim. So buyer beware when you're reading the literature. The most egregious example of this was our Brugada syndrome expert panel that curated the literature and decided that one gene was definitive and the remaining 20 genes were disputed. So, and if you look at why might this happen, this is actually not from the Brugada group, but this is from our hypertrophic cardiomyopathy group. These are the genes in the limited and no evidence categories. And what you often see is there'll be a tiny bit of genetic evidence. The genetic is in red. And then most of the supporting data is functional studies, which don't always really represent a good model of disease. And those are times when things go awry. And I can tell you from the Brugada syndrome example, it was a situation where these were ion channel genes. They had the one SCN5A, I think it was, that is the valid model, and they could show disruption of the function of the ion channel was correlated with pathogenicity of the variants. And there were a lot of human examples for that particular gene, but then they just used that ion channel model for all of the other ion channel genes and said, oh, if we disrupt the function of the ion channel, we can therefore assume that that would cause a Brugada syndrome phenotype, a short QT arrhythmia. And that is not necessarily a valid assumption to extend that just disruption of the ion channel, therefore, is actually correlated with phenotype. And the only findings in humans were just a bunch of missense variants that had no other evidence. So that was where the sort of whole assumption broke down. And ultimately, the work of that Brugada syndrome group included the authors of those original papers who did agree that data was not sufficient. So, so it's useful to get experts together and really think about the evidence in a very structured way. So if you want to look up your favorite gene and whether it's been curated, you can go onto the ClinGen website, and this is just some screenshot of it. You can search the all curated genes tab and see whether any curations in the different domains have been done. So MYH7 has been curated in most all of them. Or you can just go specifically to gene disease validity if that's the domain you're interested in. You'll get a full list of everything. You can export it, you can search on it, you can do whatever you want with it. All open, licensed, free to use as you wish. I will point out, so now I've clicked on one gene, in this case, ACTC1, and now I'm showing the detailed curations that have been done for gene disease validity, dosage sensitivity, and clinical actionability. And each of those green buttons, you can click on it and open up an entire detailed description of the evidence that was curated to support that claim. But one thing I want to point out here is that for any given gene, it can actually be associated to multiple distinct diseases. So in this case, ACTC1 has been implicated in both hypertrophic cardiomyopathy, dilated cardiomyopathy, and arrhythmogenic right ventricular cardiomyopathy. But one of those is definitive, the other is moderate, and the other has essentially no evidence that could be curated. So you can see, some claims can be definitive, but others for that gene may not be. And the evidence gets curated separately for each of these diseases, where you can come up with different interpretations. One thing I did want to point out, there's been a project called the MAIN project, which stands for Matched Annotation from NCBI and MBLEBI. And this has been a really critical effort to get the two major resources of gene transcripts to be aligned and have the same sequence, which has been a headache for our community for many years. And so that group has not only made sure, so they define the main select transcript, which is the primary default reporting transcript when you're describing variation for that gene. And there's only one per gene. That does not mean there's only one biologically relevant transcript for any given gene. It means they have gone and selected based on biological data a transcript that they believe is the most biologically relevant transcript for that gene. And then that gets tagged as the main select. Now, they also will tag what's called main plus clinical, and that is additional transcripts for the gene for which there is exon or non-coding UTRs that has known pathogenic variation that is absent from the main select transcript. That way, if you're offering a clinical test, you can ensure that you're covering all of the regions that could harbor pathogenic variation. Now, again, it does not mean that there are no other biologically relevant transcripts beyond these two sets. It's just no pathogenic variation has yet been found in other transcripts to get them into one of these two lists. These all match build 38, and they must be well supported, conserved, expressed, et cetera. So when you find variants, you first see if you can report them on the main select transcript. If the site is missing from that, you move to main plus clinical. If it's not on that, then you use whatever best transcript you find. Okay, and main select is now incorporated into all the major genomic platforms like ClinVar defaults to the main select, Nomad, et cetera. So use it if you aren't already. Now that gene disease validity work, so I told you the guidance from ACMG on how to use that work. So is it actually happening in the clinical community? Yes, this is Invitae's panel for Brugada syndrome, which used to have 21 or so genes, 20 genes until that paper came out. And then they changed their default panel to one gene, that one that had definitive evidence, and they shoved them all into this, the rest into what they call add-on preliminary evidence genes. So a physician has to actively go in and say, I want to include these genes that aren't yet definitively incorporated. So that's sort of how Invitae deals with this. Other labs deal with it in different ways. Okay, and again, feel free to ask questions along the way. But now we're going to start transitioning into variant classification. And one thing I want to point out that I sort of alluded to at the beginning is that if a gene scores in the categories of limited, no evidence, distributed, refuted, all variation in those genes must be classified as VUS. You actually can't even classify it as benign or likely benign, because again, that implies an association with disease for which it is not associated, right? So it's just uncertain. What we kind of do is, if there's a gene with moderate level evidence for the gene disease validity relationship, we will cap it at likely pathogenic. And so you might find a clear loss of function variant, but its lack of full confidence is actually related to the gene disease relationship not be fully there, even though the disruption of function is maybe clear. So that's sort of how we correlate the gene disease validity and the pathogenicity. And then the term pathogenic can be used in genes that are strong and definitive. Well, okay. All right, I mentioned this other database that we've started called the GenCC. GenCC stands for the Gene Curation Coalition. And basically, a number of years ago, we recognized that there were multiple gene-level databases out and about: OMIM, Orphanet, the TGMI G2P, the Genomics England Panel App, Australian Genomics Panel App. These are all public databases that have relationships between genes and diseases defined. And those databases were all over the map in terms of the terminologies and ways they were showing relationships. Some used color systems. They were the opposite. Red was implicated in disease in one database, and red was not implicated in disease in another database. It's just a mess. So we got all these groups together and said we need to align all these resources, use common terminology. We went through a whole Delphi survey process to define the terms, which in the end ended up being pretty much the ClinGen framework that I already shared with you, although we added one other category that was animal model only. So really systematically defining those genes that are clearly implicated in a non-human phenotype, but not yet implicated in a human disease. And we have a special category for those now after this work. But we launched this GenCC database, and we also recognized that there were a bunch of clinical labs that were doing all this gene disease curation also, in some cases using ClinGen's framework, just to decide which genes should go in their panels, and they were willing to publicly share that curation. So we got all of those groups on board, and everybody involved is up across the top, and we have a steering committee with representatives of all those databases. And so now We put all of this data into one database called the GenCC database. And there's a paper and a podcast if you want to hear more about it. Marina DeStefano in our group has led most of this work. And so here is the sort of interface for searching for a gene. So you can go in and search for your favorite gene. And when it comes up, you can click on it and expand and quickly see who's submitted and on what condition and what their claim was. There's also a little summary button. I will say we had to add a new category called supportive. And we use that for two databases that really don't delineate the evidence. One is OMEM and one is Orphanet, and they just have a binary. It's in the database or it's not, but it doesn't really correspond to all these delineated categories. We just shove them in supportive for those two databases. But so at least you can see what's there. And we're soon to integrate the OMEM database. Is not quite there yet. They're waiting for an API-based submission, an automated submission, to ensure that GenCC is always in sync with OMEM. And then what we also do now that this is the benefit of ClinVar. Everybody submits, and then you can see where we disagree. In this database now, everybody's submitting, so we can quickly see where we disagree with gene disease validity classifications. And so then we bring them up. We have monthly meetings for GenCC. Marina presents the evidence and gets the evidence from each of the groups, and they share their evidence. And then we delineate whether really just in a binary approach, should it be moderate and above or limited and below? Because that's really that boundary between included in a clinical test or don't. And so we don't necessarily debate, is it moderate or strong? We just say above or below this line. Occasionally we hit genes that are just so on the border that we just leave them at borderline. So that's what we've been doing. We've now resolved 32 discrepancies, and Marina has a nice list that she maintains of all of the discrepancy resolutions. And then we're going to work on GenCC actually being a submitter to that database so that we can show when we've resolved a difference. Okay, so that's everything I want to say about genes. Now I'm going to move to variant classification. It's a lot more involved. We will never have experts be able to classify every variant found in the human genome. There's billions of them. So we have to take a different approach. One of those is to develop common standards and disseminate them to community, hoping that everybody out there will use the same standards to classify variants. And then we will have more robust databases, because we can't possibly get experts doing this at a rate of a few variants a month to ever tackle all of this. So we put out standards, and those first standards that have been really more rigorous came from work we did with American College of Medical Genetics and the Association of Molecular Pathology. That guideline came out in 2015. Here's our group drinking wine because it was really stressful to develop these guidances. It took us a while, but we eventually came out with a framework. And I just want to say, when you think about variant classification, there's different ways to think about it. So one, you might be asking, does the variant impact gene or protein function? And I told you early on, you can't call a variant pathogenic if the gene isn't implicated in disease, but you can say it may disrupt function if you know what the function is. But when we talk about variant classification, that is this step. Does the impact of this gene or protein lead to a disease phenotype? Is it associated disease? So variant A is pathogenic for cystic fibrosis in an autosomal recessive manner. So you're really defining the clinical significance in the context of a disease with an inheritance pattern. And that is what we developed the standards for. But a lot of us do this work for the purpose of the third step, which is we get a patient in hand and we're trying to say, does your thickened heart, is it due to this pathogenic variant in MYH7 that's associated with hypertrophic cardiomyopathy in a dominant manner? And then we might say, yeah, the variant pathogenic, but no, I don't think it's causing my patient's phenotype, because that's a Brugada syndrome variant and they have hypertrophic cardiomyopathy. The two aren't related. So this is a different step. So I'm going to talk to you about step A, how do we classify a variant's role in disease? But keep in mind that does not, just because you find a pathogenic variant doesn't mean it's causing whatever your patient's phenotype is. That is a separate analysis that relates to correlation of phenotype between the patient and whatever the gene disease relationship is that you're talking about. We also, when we develop those guidelines, we created it. Well, I didn't point this out, but in the last slide, oops, oh dear, what did I just do? Hold on. Okay. On the left side, with that 2015 guideline, we defined 5 terms for variant classification: pathogenic, likely pathogenic, uncertain significance, likely benign, benign. And those 2 terms, likely, where there is no formal quantitative definition for the word likely. Look it up in a dictionary, doesn't give you anything. So we, by popular vote in our committee, decided to define that term for variant classification at the level of 90%. Why did we do that? Because we felt that clinicians would be willing to act on information that is at least a 90% level of confidence. That was sort of the rationale. So we defined that, and that has then enabled other studies that are quantitative. Let's say you validate a functional assay and show that it is 90% accurate for calling the pathogenicity a variant. Well, then you can use the term likely pathogenic for that assay because you've proven that it's at least 90%. Whereas if we had defined it as 55%, then those assays could use the term likely path at a very low level, which wouldn't be good. So even though much of our variant classification is not very quantitative, I can tell you, when we have quantitative assays, we can now have a target for what you need to get to call something X, Y, or Z. So that was why we did that approach. So when we came out with the 2015 guideline, we defined all different types of evidence, and then what we believed was the level of strength of that evidence: supporting, moderate, strong, and very strong for these different types of evidence. And then same thing on the benign spectrum. We had standalone. The allele is highly frequent in the population. It cannot, by definition, cause a Mendelian rare phenotype. Now, there are hypomorphic alleles, and there are times when you really need to look carefully, because there can be some alleles that can even reach 5% allele frequency. And we sometimes see them as these hypomorphic alleles that might lead to recessive disease when combined with a severe allele on the other copy of the gene. And so there's a handful of these variants that, despite reaching reasonable allele frequencies. So in this standalone, we always say you have to still check ClinVar in the literature just to make sure there's no claims of a hypomorphic allele that has sufficient evidence. But largely that can be a standalone, but the rest is sort of different types of evidence you have to bring together. And it relates to different domains of evidence: population data, computational predictive data, functional data, segregation data, de novo occurrence, allelic on the other strand with another variant, and then other data related to the specificity of the phenotype. Now, and then there's sort of a combination menu. If you have one of these and 2 of these and this and that, it gets you to your classification. So that's how this framework was defined. And I don't have time today, with only 10 minutes left, to go through all these different types of evidence and how we apply them, but we have lots of courses to learn how to do this if you're interested in taking that next step. So in addition to this framework that we came out with in 2015, we then immediately formed our own sequence variant classification working group within ClinGen, really to evolve these standards. And so it's Stephen Harrison, Les Biesecker co-chair our group. We have been continually refining and evolving these evidence-based standards. And then we also have all these expert panels that are classifying variants in their particular genes of relevance. And they come up with specifications of the standard. So this is the allele frequency threshold for this gene and disease, that above which we would call it benign, and below which this is a functional assay that's valid, this one is not. So they sort of define all the things that are relevant to their genes and disease. And this group then reviews those proposed to make sure none of these groups are going off the rails with what they're doing. And then we've been gradually trying to move this framework to a more quantitative basis, as opposed to supporting, moderate, kind of vague terms. So just as an example of that, for the PS2 and PM6 de novo codes, we then defined a point-based framework defining how specific the phenotype has to be for the gene, and that correlates to how many points and how many probands? Was it seen de novo in one individual or seen de novo in 12 individuals? That's obviously very different evidence. And did you check and confirm that the parents that didn't have the variant were actually biologically related to the proband? So there's different things if you did confirm that, or you assumed that, but didn't have parental STRs or exome trio data, for instance. So that correlates with how all these points are assigned and how specific, is this a patient with autism and you found a de novo variant in Titan? Not a very specific relationship versus a very small gene with a very small target and a very specific phenotype of Gorlin syndrome and this gene, and that's more evidence. So all that sort of phenotype framework. And then based on the sum of the points, then you can correlate with these different strengths. So that's a lot more detail about how to apply this rule than just saying, if you find a de novo, apply this code. So that's the kind of thing our group has been doing over the last few years. And on the ClinGen website, under the sequence variant interpretation work group, are all the different sort of more detailed guidance for how to apply these different rules that we've been working on over the years. I can tell you, though, that we reformed the ACMG work group. It's now a joint work group between ACMG, AMP, CAP, and ClinGen, all 4 groups working together. And we have moved the system to a point-based system. And we've added sub-tiers of the VUS category. So the VUS was anything that was like between 90% likely and 10% unlikely. But that's a big range, right? And some variants are like, oh, they're almost benign, just don't pay attention to them, versus, oh, we just a little bit more evidence, it'll be likely passed. So it really makes a difference to know which end of that spectrum you're on when you give a variant back to a physician and a patient. Do they take the time to do other follow-up studies? Do they bring in family members for segregation analysis? That's work, cost, expense. And you probably would do it if you're on that side, but not on this side, right? So we're adding that into the system. And when it goes to a point-based system, it'll automatically tell you which sub-tier you're in. You don't have to even think about it. So that's sort of the basic kind of framework and approach we use to classify variants without getting into all the details of how we apply that evidence. But as I told you before, we can't possibly have experts classifying every variant. So we rely on crowdsourcing the problem also. And years ago, we worked with NCBI to help guide them to develop the ClinVar database and then work with everyone in the community to get them to submit to ClinVar. And honestly, I never thought it would happen, but it did. Now I did have to pay them subcontracts in my first ClinGen grant to get it off the ground, but I no longer pay anybody and they still submit, and it's brilliant. And we have over 3 million variant submissions on over 2.3 million variants from 2,665 submitters from 90 countries. Brilliant. And most of them are data coming from clinical labs, because that's where we focus, is getting those clinical labs that we're doing high throughput, systematic classification of variants and getting them to share that. There is some data from researchers and publications, some data from our expert panels, some data from patient registries and clinics, but vast majority of the data is coming from clinical labs. And we developed this STAR system for ClinVar, where, because we're like, some people are like, it has to be a clinical grade variant database. Only the things we absolutely know can be in it, so we can just use it for clinical care. And I said, that's great. We'll have 10 variants in it. Because most of the variants we see have been seen before or have very little data, and I still have to sign out a patient report. And I want to know any evidence from anywhere in the world to help me interpret that variant. So we said we should let everybody submit, but we do want to define when something's a little more reliable than other things. So we have this STAR system. And if you don't tell us how you classify variants, yes, question. Oh, 5 minutes, thank you. If you don't tell us how you classify variants, you get 0 stars. OMIM is in that category. They don't actually rigorously classify variants. They just dump them in from the papers, so they get no stars. But then most of the clinical labs, they have criteria for how they classify variants. They largely all use the ACMG AMP guidelines, and they get a star, and they override any of the 0 star submissions. Then if everybody who submitted something and there's more than one, and they all agree, it gets 2 stars. And then if an expert panel has classified it, it overrides all of this and it gets 3 stars. it's in a professional guideline, which is actually very few variants, it gets 4 stars. So that's sort of the system. This also allows researchers who want to use that database in aggregate to say, I'm only going to use the 3 and 4 star group, or I'm going to only use the 2 and 3 and 4 star group. And I'm going to ignore the 1 and 0 star, or I'm going to use the 1, or look at my data when I use this versus look at my data when I use that. So it allows that sort of aggregate with some degree of confidence. It doesn't mean that everything that's a 2 star is right and everything that's a 0 star is wrong. It's just, you know, a little way to help make it a little more accurate. But by all these labs submitting, we can also then identify interlab conflicts. You look at the database. So first thing to point out, oh yes, question.
Do you also have a system to downgrade if you have a conflicting information from
So
someone
we
else?
just launched, will actually be in the little, actually it's on our website. We've launched a new system because there are things in ClinVar that we know are inaccurate. And I can go in and be like, that's wrong. Sometimes you'll have variants in there with 40 submissions and 39 say pathogenic and 1 from 10 years ago says uncertain. Like, and it just causes a conflict. And so we now have a system to go in and we don't remove them. What we do is we flag them as candidates for removal. And our ClinGen team at the Broad here does that work. And it's usually fairly objective, like it's pretty clear that it's wrong. And so when we flag them, we then send the list of all the flagged variants onto ClinVar, and anyone in the community can now nominate. We have a little nomination form we just launched on our website. You can say, I think this variant's wrong. Can you look at it? So we'll go in, if it's clearly wrong, we will flag it. We will send, and then monthly we send the list over to ClinVar. They then send an email to all the labs whose variants have been flagged and say you have 60 days to update it or remove it. And if not, it's going to get flagged. What does flag mean? It means it no longer contributes to the overall classification, but it's still left there in a flagged state because sometimes there's evidence in there. It was seen in a patient. It's just that you shouldn't have called it pathogenic because you just had 1 patient. But if you combine that with the others, you might be able to build your evidence. So we leave the entries in there, but they no longer contribute to the overall classification in ClinVar. So that, does that get at your question? We have a way to do this now, which has been a pain point for a while. Okay, but I do want to point out 76% of variants in ClinVar come from 1 lab. It just emphasizes the daunting thing that we have here, which is that most variation we interpret and report in patients, you get 1 shot at it, that's it. Luckily, in some cases, we see the variants over and over again, and we have more data. And in ClinVar today, there are about 1% have what we call medically significant differences, where one group is calling it path or likely path, and another group is calling it either VUS, likely benign, or benign. And that changes patient care. And so we really focus on tackling those. And actually, this ClinVar curation project we're doing is tackling all these medically significant differences to see where it's real differences, people disagree, versus just old things mucking up the system we're going to take out. So we're working on that. Okay, I only have 0 minutes left, so I'm just going to fly through the very end. We do discrepancy resolution between labs, and then I told you we engage experts. We have 110 expert panels around all these different disease areas, people all around the world working. They have to go to a rigorous process to develop their specifications. When they do that, all our expert panels get the FDA recognized tag. This is a summary of all the work. It's on links. I'll send you my slides. If you want to volunteer and get involved, it's a great way to learn more about this. On the website, there's a sign up form to get involved and be assigned to one of these expert panels. You'll learn much more about this. Or if you're an expert in a disease area, you can also tell us and you can join a specific expert panel that relates to your expertise. This is the wonderful group of people who lead ClinGen from lots of people around the world. And we also have a curation interface. So if you're a researcher and you just want to use the ACMG guidelines, you can freely use this interface. Our Broad Rare Disease group uses it for all our work. And there's just some screenshots in there. And one thing to point out, if your gene is limited, we have ways to address that through the Matchmaker Exchange, but that's for another talk. But it's a brilliant system, and you can read about it if you get my slides, and I'll stop it. Okay.
There was one question in the chat, and it was actually related to a broader one I was interested in, is that thought of how to predict the deleteriousness of missense variants. And there's obviously many different scores available. Everyone's now excited about alpha missense.
Yes.
Do you use any of, what's the pace at which new predictions are integrated into your consideration?
Yes. So in the original ACMG guidelines, we had a category for in silico predictors. Largely at that time, it was things like polyphen and CIF. Now there's a lot more of them. There's meta predictors like Revel and other things. And so at the time, it was relegated to supporting and not anything stronger in terms of how you use this evidence. It wasn't believed to be very good in terms of really predicting. However, over time, those predictors have actually gotten much better. And now SVI has published a paper giving additional guidance on how to validate these predictors and the standards for that validation, and showing that for some of these predictors with certain scores, you can get up to as high as strong level evidence. There's been some look at alpha missense, which just came out, and it's looking like it's not that much better than Revel and some of the other good predictors. So it's on the better side, but not really like a leap above the existing meta predictors that are out there. And actually anodonoluria and I think Sarah Stetton and her group are doing some look at sort of for the average person, how many missense variants in their entire genome actually would reach a level of strong, to give some like, is this out of whack or does this make sense? And actually that data is looking pretty good, that very few, like 1 to 2 per person, reach that strong level to really feel like, so they're better, they're evidence, we use it, it's part of the guideline, but I can tell you none of these predictors are 100% accurate, and they have fail modes. And also each gene is different. You really have to validate it per gene. And for some genes, they work better. For other genes, they don't work well. And that may relate to the gain of function versus loss of function and a variety of different things. But so short answer is yes, we use them. We have guidance for it, but they're not perfect. Yeah.
I haven't fully thought about this question completely to formulate it well, but given everything is based on GRCS 38, now we have CHM 13. How do things change, and how do you interpret specifically for those regions which are completely novel?
Yeah.
And how do you think about what needs to be changed from both the ends, from the ACMG angle and from researchers' angle, to find new evidence?
Yeah, so we are definitely trying to really get the community to move to Build 38. It's been 8 years, it's time. And Nomad will finally launch. Nomad version 4 will launch on Wednesday next week to start ASHG. It will now be all on Build 38, which we hope is another reason to move, because the number of clinical labs out there today that aren't on Build 38 alone is shocking. That said, it's kind of made a little bit of a mess in that now you have these other genomes and the pan genome reference coming, and all of this. So there's a lot of discussion around how to support. Though the other genomes are not in a state for the community to switch to them. There's a lot of work to be done. So we're really trying to get everyone to move to Build 38 right now. However, there's a lot of work being done in long range, long read sequencing that are starting to define these regions of the genome that we've had largely inaccessible to date. The challenge is that the population variation is not defined that. So Mike Talkowski's just submitted a grant 2 weeks ago, written literally in 4 days. Once the program officer said he could submit a grant, he wrote it in 4 days, and it's to build a version of Nomad with long read data so that we can start to get population frequencies in these regions of the genome that we have had that have been inaccessible. So we also need better analytical tools to use long read data to look at these regions. So there's work being done to take advantage of these new approaches, also to fix some genes that are problematic on the current Build 38 even. So more work being done, but the field is gradually making progress. I don't know if that totally answers your question, but where it is. Other questions? We have 3 minutes? Go ahead.
So I love the idea of main select because it helps to, so I'm on a congenital heart disease panel and everyone's mapped their variants to the transcript that predicts the best pathogenicity regardless of its relevance. Mm-hmm. Have you thought of extending that to like developmental main? Because when we look, there's a lot of congenital anomalies and other early onset disorders where GTEx might not accurately represent, or other large references, the real transcripts that are most relevant to our curation topic.
Yeah, so a couple things happening. So the main group that spans EBI and NCBI, they use a lot of different resources to understand the biology, where transcription starts and stops, and to try and inform this. But sometimes they don't have all of the evidence, and there's other evidence out there. And as you said, getting early development stage transcriptional information is important. And one transcript might be relevant for one disease and a different transcript for a different disease, right? So the ClinGen expert panels are actually doing some of that work. So they may define, for our disease that we're curating, this is the right transcript and that is not the right one. Also, there's been a tendency like, oh, my variant's in the main select, so therefore anything will disrupt function. Like, just because it's in the main select doesn't mean that there's not an alternative transcript that's still functional, missing a couple of exons. And that is true of BRCA 1 or 2, I forget which one, but there's like 2 exons that are non-essential, and loss of function variation in the main select transcript in either of those 2 exons does not lead to disease. So though the expert panels define that, and they say loss of function variation in these 2 exons cannot be interpreted, 0 points for PVS1, and they'll give that guidance. So it is work. So one thing, there have been some main select that were just wrong, and the experts came with data and they actually changed the transcript in main select. So if you actually believe the transcript is wrong, you can contact them, give your evidence, and if they agree, they will change that. But sometimes it's just a matter of use this one for that, use that one for that. In that case, you can say, can you add this as the main plus clinical, and then this is when it should be used versus that should be used, and define that. And we keep track of that in the ClinGen side. So yes, good question. Great.
The scale of this may be too huge to consider, but especially with that BRCA evidence where, there are modifications to how you would apply variant curation evidence based on exon.
Yeah.
Has there ever been any thought or effort given to the idea of actually doing expert variant curation, at least for genes with actionability, where it could be a really significant impact if a lab wasn't curating variants according to some of these more nuanced curation rules, especially those that have come out since the original publication, and modify how we would apply evidence?
Yeah, so we have, 65 expert panels doing variant curation for sets of genes, and they span different disease areas. And those experts include both clinicians, disease-specific researchers, also representatives of all the clinical labs that are offering genetic testing. And often the clinical labs will bring evidence from their testing in, the clinicians and researchers bring evidence or expertise from functional, and that together they define the rules for variant curation. We have a higher focus on some of the more actionable genes to ensure they have variant curation expert panels, so that the variants that are most relevant to our population are actually getting curated. So there is definitely prioritization by gene and disease based on actionability. I don't know if that's getting at what you're asking about. Great, yeah. So, I mean, it's obviously a huge pile of work. There are, thousands of variants and hundreds of genes that are actionable, and, we're just starting in some of them, but you got to start somewhere. So you should all sign up to volunteer to be curators for our expert panel. All right, with that, I think we're out of time. So thank you.